Hello, my name is Mizuki and I'm based in UKDR in Cardiff. And then this is my poster about exploring the impact of high endocytic polygenic risk in late onset Alzheimer disease using iPSC derived microglia. So what my research is about is that focusing on the endocytosis, which is the essential cellular process to take stuff in or take stuff out. And then recent study discovered that a lot of risk genes associated with Alzheimer's disease are related to the endocytic pathway. So what we have done is looking at those risk score in Alzheimer's disease and then non-Alzheimer's disease patient, and then we derived the iPSC from the PBMCs and then derived into microglia. So in total, we got 20 patient cell lines and 12 healthy control cell lines, and then differentiate into microglia, and then we looked at functional differences including endocytosis and lysosomal and all other functions. So this is the selection criteria, and then these are the polygenic risk score that we calculated, and then they are free from ApoE, meaning that everything has the ApoE3 background, so any of the endocytic pathway that we looked at is independent from ApoE allele, which is good. And then once we got those cell lines, reprogram into the iPSC, we differentiate into the microglia using the Washer protocol. Yes. And then these are the quality control we have done. So we looked at those pluripotency markers when they're iPSC, and then when they formed the EVs, they are nicely formed EVs, and then we looked at the microglial marker once they're differentiated. So they are differentiated in the microglia, and then they express those markers that are abundant in microglia. So when we look at that, we checked sort of 6 different functional, so mitochondria, phagocytic activity, endocytic activity, and the lysosomal activity, endolysosomal interaction, and also lipid droplet. We are currently analyzing those data, but here is our very, very parallel result, looking at the amyloid beta endocytosis. So what we have done is we got the control cell line and high polygenic risk cell line, and then we either treated with LPS to activate them or without it, and then looked at how much endocytosis occurs in amyloid beta when they fed them. And what we found is that there's no difference so much, but LPS significantly reduced it. So what we are doing now is to expand to the bigger process and then into further details.